All right. So we're now recording. Again, welcome everyone. Those of you who have joined us so far. This is the Biomedical Research and Mentoring Program. This is the Mentor Meet and Greet. So over the course of the next 2 hours, you will be able to hear from all the mentors for this year. So I'm incredibly excited to go on this journey with you for the next 2 hours. And there are a few housekeeping rules, and I'll probably circle back to these one more time right before we have our first presenter speak, because I think more people will be jumping on in a couple of minutes. But please, to try to keep the bandwidth of the call down, please turn off your mics and your video, and the presenter, our mentors, if you would like to turn your video on, if you're comfortable with that, I think that's great. But essentially, you should be the only person with that video on. I'll be running the slideshow, so you'll also hear, the mentors asking me for the next slide throughout this presentation. In terms of the timing, students, you should see that most up-to-date schedule posted to the RaMP website through honors. I also have a copy of that schedule in our very first, our second slide. So I'll fire up that presentation in just a moment so you all can see that, that presentation plan. Outside of that, remember that these are relatively quick, high-level overviews of the mentors, of the work that they're doing, their projects, and what they're looking for you as a potential mentee, student in their lab. So the mentors will only be talking for about 2 or 3 minutes. And the next part that I'll say, students, I know you might be dying to ask certain questions. Please hold all of your questions. Even when it comes to the chat, hold all of those questions because we want to provide the proper respect for the mentors that are going. There will be more than enough opportunity. I know many of the mentors today have posted their availability in breakout rooms, whether it's through Zoom or WebEx or some other means. That is also up on the RaMP website under project descriptions. So if you haven't already looked through those, please look through those and maybe follow along as you're listening to the presentations today so you can strategically choose which breakout rooms am I heading to once 5 p.m. hits. So that would be my advice to you students, and that's the best way to follow up with any specific questions that come up through the course of this. Outside of that, I know that some mentors have graciously included their emails and encourage you to email them with your questions if they're not available to do the breakout session at 5 p.m. I think that was everything I wanted to cover. Let me start sharing our presentation today, and I will show you that schedule before we hear from our first mentor. All right. So here's the presentation schedule. So we will be getting started literally in 1 minute. We will be adhering to the schedule as best we can. We could potentially go a little bit faster depending on the length of the presentations, but this should be a pretty good idea of what you're looking at. In the interest of time, I will not be introing mentors. I will leave that to them so everything can be streamlined. And with that, I see we just hit 3:05. So we will hear from our first presenter today. And I'm glad to see you all here. Enjoy.
And good afternoon, everyone. My name is Zakia Abdelhamid, and I am a research associate in Dr. Stockman Laboratory in the Division of Human Genetics, Cincinnati Children's Hospital. And here is the link for our lab, so you can check it and you can see the projects that we are working on the lab. So we have so many projects that you can look them see if any one of them other than this one that I'm presenting today that you are interested in, you can also look this up. Today I'm gonna talk to you about, or the project that I will, if you join the lab, will be investigating the role of the primary cilia in the developing, during early embryonic development in the fallopian tube. And next slide, please. Next slide. One more. Click, please. Can you click one more? Yes. Here you go. Thank you. And there are two types of cilia in the mammalian body. Primary cilia that you can see them in all interphase non-dividing cells. And there are another type of cilia that have specific tissue localization. It's called the motile cilia. And we see these either in the respiratory tract or in the brain ependymal cells and the fallopian tube cilia, and also some ducts in the male genital system. However, all what we know all the years is that the fallopian tube cilia is motile cilia, but until recently we get to know. Can I have the next slide, please? We get to know that the fallopian tube epithelial lining is also not only have the motile cilia, but also there is secretory cells that carry primary cilia, which is very important signaling hub of the cell. And the function of these cilia during early embryonic development, and if this cilia is defective in idiopathic female infertility, is never been explored. So this group have found it like over a decade ago, and then a couple of years back, I found it very nicely in the human patient fallopian tube. Also, you see that these bunches are the motile cilia, but the very tiny little ones are the cilia. And next slide, please. The project is about knocking out these cilia on these secretory cells specifically. And to do that, we have a Cree line that only expressed in the secretory epithelial cells of the fallopian tubes that carry these primary cilia. And you can, and it's inducible Cree, so you inject doxycycline, and you see that the expression flares up in the fallopian tube, only sparing the rest of the female genital system. And we're going to knock the cilia using the KIF3A mouse, and we are developing this line in the lab right now, and it's about to be ready by January for you to start on. And the next is
actually, can you fix the view? We're only seeing this last part of the screen.
Okay, my apologies. Let me figure out what was happening.
It looks like you need to share your desk, not your screen. I mean, you need to share the file.
Oh, okay. I think I see.
The view is
still the same thing.
Yeah.
I'm not sure why it is doing this. My apologies, everyone.
If you go under display settings, you might, so click that, the display settings drop-down menu there at the top.
Mm-hmm.
And
Yeah.
there, you can
have
be
a
able
full
to,
screen there.
Here, I am going to try.
No,
Let me try.
It's probably in the way you're showing the PowerPoint. I don't think it has to do with the. It's just hard to see the slides the way it is.
Yeah.
Yes, like
At the bottom, you can see the fourth one. Kyle, at the bottom, you have to select the fourth one, just next to the. Yeah, display settings, the fourth tab after display settings. Just next to where the cursor is.
Okay, sorry, I'm trying to follow. I promise I've done this successfully before.
The bottom of the screen there, you can see there's the one that's got the little
Right here?
Yeah.
right next to
Okay.
the size. Turn over.
On the right
keep,
side.
Yeah,
Go to the right side. One,
over.
two, three.
Keep going, keep going, keep going. There you
Yeah,
go.
that
Click
one.
on that. That should do it.
So see, the problem is it's showing that full screen slideshow, but it's showing it on my other monitor. It is mixing up my monitors when I'm sharing for some
It's
reason.
just switch monitors.
All right, I'll try to do that.
2 monitors. It's only sharing one of your monitors.
All right.
If you click the display settings at the top of their PowerPoint file, there's 4 different tabs. Hit the display settings, and it'll allow you to swap your which monitor you're viewing it from.
All right, 4 different tabs at the top of my PowerPoint. When I'm presenting or when I'm not presenting?
Yeah. So go ahead and share your screen like you did before.
Mm-hmm.
I've done that. Yeah, I ran into this problem quite a few times when I was lecturing. So. All
right. So if I'm sharing.
Go ahead and present the PowerPoint like you had previously.
Okay.
And then do you see at the top, it says show taskbar and then display settings?
Mm-hmm.
So under display settings, hit swap presenter view and slideshow.
Okay. Is that working for everyone now?
Success.
Yes.
Thank you so much. That's the value of collaboration right there.
You are welcome. Yeah.
Thanks, Dimitri.
All right. So so this works out well actually because I have built in some time if we were running over at all. So let me get to the slide we were on so we can continue hearing from you, Zakia, and my apologies once again. Do you want to start off from this slide or the next one?
Next one, please.
Okay.
Okay.
So the aim of this and the proposal of this study would be to functionally characterize the importance of the newly discovered primary cilia on the secretory epithelial cells of the fallopian tube. And we want to understand more about how the function of these cilia contribute to early embryonic development. And this also can be translated to get more information or more data to explain some cases of female idiopathic infertility. This is our ultimate hope out of this. And during this training, you will be trained, the mentee will be trained on the animal husbandry and mouse genetics, lots of mouse genetics you will be going through, and also histology, BCRs, and confocal microscopy, and also probably some super resolution radial fluctuation imaging that can be done also to stain for cilia or to stain certain antibodies or certain proteins in these primary cilia as a project permit. And thank you very much. Thank you.
If you can, yeah, perfect. So good afternoon, my name is Raquel and I'm a postdoc in Dr. Kimba's lab. We are in the Division of Pulmonary Biology and Neonatology in Cincinnati Children's. So in our lab, we have different projects, one like related to non-clinical or basic science, where we work with lungs, specifically with lung surfactant protein D, in the context of in the innate immune response, pulmonary and extrapulmonary infections. We also have some translational projects going on in which we have a collaboration with a pharmaceutical company or with therapeutics that provide us a recombinant version of this protein, and we do some functional assays for them. And last, we also have some clinical projects in which we work with data from the babies. So if you can go to the next one. So the project that we are proposing for the RaMP student will be in the area of the basic science and translational science and working with surfactant protein D. So SPE has been very well characterized in the area of the respiratory system. And we know that the protein can bind viruses and bacteria, and that binding is going to help to eliminate those pathogens from the lungs. We also know that it modulates the inflammatory response. However, what SPD does outside the lungs is not very well explored. So we have a project in which we are studying the role of SPD in extrapulmonary infections, for example, in an animal model of sepsis. And the student that gets to the lab will get a sub-project inside this big one. So the techniques that they will learn to work with animal mouse models and also some molecular biology and biochemistry techniques, such as to identify and quantify protein concentration, for example, ELISA and Western blots, cell quantification and identification of inflammatory cells, and some other histology techniques. Next. So as a member of the lab, you will learn how to work in a research lab environment, the techniques that we just mentioned, to design experiments, record data properly, analyze the data, and also how to present that data as results. So on the right side, you have like the characteristics that we are looking for in a student. And I just want to highlight that we don't require prior lab experience, and that we have flexibility to come in the lab like 2 days more hours rather than coming late. And that if you have further questions, I will be in this Zoom meeting room from 5 to 5:30. Thank you.
Let's see. Hi, everyone. My name is Madeline Bonfield. I'm a third-year PhD student in the immunology program, and I'm working with Dr. Hitesh Deshmukh. And broadly speaking, my project investigates the link between the environment of the developing newborn lung and innate immune responses. And specifically, we're looking at a type of innate lymphoid cell called ILC3s. Next slide, please. Our lab is focused on understanding and improving newborn health, specifically looking at immune responses within the lung. And we're interested in how early life experiences shape the development and health of the newborn lungs, with focus on how antibiotic exposure before or shortly after birth can alter the microbiome and how that then affects lung immunity. Our lab has identified a specific cell of the innate immune compartment, that's those ILC3s I mentioned earlier, as critical in the defense of newborn mice against pneumonia infection. Sorry, guys. And then my project focuses on understanding how those cells protect the developing lung. And as a quick note, something came up and I'm no longer available to meet by WebEx today. So please, if you have any questions during this talk, shoot me an email, or if you want to schedule another time to meet later. Next slide, please. So as I mentioned, I'm looking at how cells are specialized to their environment with a focus on ILC3s in the neonatal lung. I like to think of this interaction in terms of a radio and a broadcast tower, where the radio represents the cell receiving signals from its environment. So for example, if I want to listen to Splendid Table on NPR, there's a number of steps that I would have to go through in order to do that. I have to turn on my radio, make sure it's tuned to the right station, make sure I'm listening at the same time the show is actually broadcast. And if any of those steps are missing, I'm not going to be able to listen to my show. And so similarly, if you're looking at a cell, there's a number of mechanisms that are allowing it to tune in to its environment. And I'm investigating a specific transcription factor, PPAR gamma, which we believe coordinates signals from the developing lung with the development and/or function of these innate lymphoid cells. Next slide, please. Okay. So if you're interested in my work, there's 2 projects that I would love your help on. And the first is an in-person bench work style project, while the other is computational and could be done remotely. And so you could choose between these projects based on what you're available for. So for that first project, I'm doing a number of experiments trying to see how depleting that transcription factor that I mentioned, PPAR gamma, affects ILC3s. And so for one of these experiments, I'm using chemicals that would inhibit or activate this transcription factor on the cells in culture. And there's a number of steps involved in this, so depending on your availability and interest, you could choose one or more of these steps to focus on. And this project uses a number of what are considered basic wet lab skills, so you'd have a great opportunity to learn and practice these skills, while also being an enormous help to my project. And then that second project would be helping me to analyze single-cell data that we're currently generating. I have basic coding knowledge and background and could help you with those basics, but we would be learning and working on applying those more advanced coding techniques together. And so this should be doable remotely. So if you really like the lab or my project, but don't want to or unable to come in and do an in-person project, we could make this one work. So thank you all for your time. And again, if you have any questions, please email me. Thanks, Kyle.
Hi, everyone. I'm Jennifer Bourne. I'm a postdoc in Dr. Susan Waltz's lab in the cancer biology department. I work in the Waltz building, so that's just kind of a disclaimer. I am on the medical campus, so just keep that in mind. And the project that I'm working on is investigating RON signaling and how RON suppresses innate immune responses in breast cancer. Kyle, next slide. So this is just an intro to the Waltz Lab team. Dr. Waltz is the PI. I'm the single postdoc in the lab. We have one graduate student. He's in his 4th year, Brian. And also Carissa just joined us over the summer. She's a research scientist and a technician who greatly helps us with our mouse animal husbandry work. And again, I included these pictures because this is what you will probably see if you come work in the lab. Next slide. And so the Waltz lab studies the RON receptor tyrosine kinase. And so previously they've discovered using multiple mouse models, both in prostate and breast cancer, that RON signaling actually drives aggressive breast cancer phenotypes. And so what they previously found is that RON drives breast cancer tumor cell growth, metastasis, as well as escaping cell death. And the role that I'm working on and that you would also be working on is learning the mechanisms to how RON actually promotes a pro-tumor microenvironment. So what we've found is that when you have high RON signaling in breast cancer, that there is actually a pro-tumor immune response. So what that means is that all of the immune cells that come into the tumor microenvironment, they are pro-tumor phenotype rather than anti-tumor. And this aids in the progression of breast cancer. And so you guys can read on the summary in the document that Kyle provided, but that's the gist of the project that you guys would be working on, is evaluating the role of RON and how it relates to the immune responses. Next slide. So that entails a lot of wet lab work. So we don't do a lot of computational work. So just as a disclaimer there. So some of the skills that you would learn in the lab, multitude of molecular biology techniques, including cell culture, Western blot, real-time PCR, immunohistochemistry, and microscopy. We do a lot of animal work. So that would require learning animal handling, as well as genotyping and learning PCR to tell if the mice are what we are actually looking for, as well as learning how to maintain mouse colonies. And this will all entail how to design experiments, data analysis. Dr. Waltz will expect you to present in lab meeting, as well as how to read and interpret scientific literature, properly maintain a lab notebook, and general day-to-day lab operations. This will go probably from bottom up. So as we gain your trust in the lab, so learning how the lab functions as a whole, more experiments and more responsibility will come with that as well. I'm not able to meet today at 5, but I did include my email and it is in the document that Kyle provided. So if you have any questions, please reach out. And just an FYI, Dr. Waltz will be involved in the interview process as well. So just keep that in mind, but everything should all be good. So thank you.
All right. Hi, everyone. My name is Nicole, and I'm a postdoctoral fellow in Aaron Zorn's lab. We're in the Division of Developmental Biology at Cincinnati Children's Hospital. And our lab is a developmental biology lab. We're interested in how the digestive organs develop during embryonic development. And I specifically study how the trachea and the esophagus form while the embryo is developing. And so there are mistakes that can happen during this process, and there are babies born with developmental defects like the esophagus ending before it connects to the stomach, so we call this synatresia, or there's an abnormal connection between the trachea and the esophagus. So the babies get surgery right away, but we would still like to know what causes these defects in order to prevent them in the future. So we think genetic mutations are involved, but we're not sure which genes are involved, and they actually control this process of how the two organs develop. So next slide. So our overall goal is actually to discover new gene mutations that cause these birth defects. And our lab uses frog models to model development and the mistakes that can occur during development in human patients. And we do this because the tracheoesophageal development is actually quite similar in frogs and humans. So the way that we do this is we do CRISPR-Cas9 gene editing, and we mutate these human patient genes in the frogs to see if the frogs get these tracheoesophageal defects. And so the RaMP student with this project will learn a little bit of frog embryology and how to design a CRISPR-Cas9 gene editing experiment, how to confirm that tadpoles actually have the correct mutations that we want using PCR genotyping, and then some staining techniques to look at the anatomy of the trachea and esophagus and see if these frogs do end up getting the defects with these mutations. So next slide. And so for days and hours in the lab, ideally great if a student could come in back-to-back days, but there is flexibility. So we expect about 2-3 days a week, and about a morning or an afternoon for each day. And my schedule is very flexible. So any weekday you can come into work, and any time that I'm physically in the lab is also possible. And I have a Zoom room set up after this presentation, so I would be happy to take any more questions at 5 o'clock.
All right, hi everybody. Next slide, please, Kyle. Oh wait, I'm missing one. It's okay. I am a postdoc in the Hayworth lab at the UC College of Medicine in the Department of Internal Medicine, and we work broadly on cardiovascular disease. So cardiovascular disease is the #1 cause of death in the United States and worldwide. It's the most common cause of death that is attributed to cardiovascular disease is coronary heart disease. And coronary heart disease is just a fancy way of saying, right, it's you've got blockages in your coronary arteries or the arteries within your heart. The most common kind of presentation of this is a heart attack. So just kind of, you can look at the little schematic I have here. So a heart attack happens when there is a blockage in coronary arteries. This leads to death of the heart muscle cells, and typically, after that, we remove the blockage, and paradoxically, that actually causes more cell death to happen. So there are 2 phases of cell death that are associated with myocardial infarctions. The first is ischemia, where that lack of oxygen and the nutrients for the tissue causes a lot of cell death. And the second wave is called the reperfusion stage of cell death, or stage of injury, wherein the rapid reintroduction of oxygen back into that environment generates reactive oxygen species and ionic imbalance, causing more cell death. Next slide, please. So currently, the way that we treat myocardial infarction patients is to first remove the blockage. So either pharmaceutically with blood thinning drugs or thrombolytics, or mechanically by actually physically removing that blockage. But we don't really have a great way to target the reperfusion injury that happens in these patients. So what we are looking at, and one of our central questions in the lab, is how can we prevent that reperfusion damage in those ischemic hearts? And what we were trying to do is use something called controlled hypoxemic reperfusion. So that's just kind of a lot of big words for saying we're basically trying to reduce the amount of oxygen that gets let back into the heart once you remove that blockage in the coronary arteries. Next slide, please. So what we are using to accomplish this goal is something called acoustic droplet vaporization. So sounds pretty fancy, but it's actually a really cool concept that's recently been developed and is pretty much headed up here at UC in the Hayworth and Holland labs. And what it is we have these droplets that we synthesize that contain a compound. This compound is pretty volatile, but it's biologically inert. So when we hit it with ultrasound, this allows for the compound inside of the droplets to rapidly expand. And it's actually pretty cool because it has a really high affinity for dissolving oxygen. So basically, once we evaporate that compound and turn these little droplets into microbubbles by using ultrasound, it actually sequesters a lot of the oxygen. So by gathering up all that oxygen, we're reducing the amount of oxygen that can be delivered to that ischemic tissue to allow the ischemic tissue to adjust following ischemia. Next slide, please. So that's what we do in the lab. So what would your role be? So this picture right here is the setup for how we actually make those droplets. And you would be using this quite a lot. It's really an interesting piece of equipment, and it's pretty fun to work with too. So we figured out, like I said, how to make those droplets and how to get them to sequester oxygen, but we need to know a lot more about them before we can actually potentially use them as a therapeutic. So what you would be doing is making these micro droplets smaller and potentially more deliverable in a human or, say, a pig, which is the model that we're moving into right now. We're also looking for a way to make them more biocompatible, so size-wise, chemical-wise, things like that. And we actually want to make sure that they can actually scavenge oxygen in whole blood, because our preliminary experiments were all done in different buffers. So I'm starting up the project right now where we're looking at scavenging oxygen in whole blood before we move into the pig model. So you would really be helping me out a lot by helping me to figure out more about these droplets and what they can do and how we can modify them. So with that said, I am not available for the meeting afterwards today, but my email is on the document that Kyle provided. So please feel free to email me with any questions, and I look forward to hearing from you. Thank you.
Good afternoon, everybody. My name is Channa Galiveti, and I'm working as a postdoc at the Lunchwind Lab at UC College of Medicine. And our research work in our lab focuses on the translational potential of the exosomes in head and neck cancer. In the proposed project, we work on the exosomal membrane proteins and to be used in the early diagnosis of head and neck cancers. Cancer is one of the most serious global health problem, and head and neck cancer is the sixth most common cancer globally. Thank you, Pat. It affects more than 800,000 cases globally and leading to more than 400,000 deaths. One of the concerning aspects is approximately 2/3 of these diagnosed cases are diagnosed at the advanced stage, leading to higher mortality and lower overall survival rate. So this emphasizes the urgent need for the identification of biomarkers that facilitate early detection and saving more patient outcomes. Next slide, please. As I mentioned, we work on exosomes in our lab, and the question is, what are exosomes? So exosomes are extracellular vesicles that are communication tools and secreted by virtually almost every cell type and present in all body fluids. And exosomes carry a lot of proteins and RNA molecules, and in the proposed project, we aim to take advantage of these exosomes that are presented on the surface of the exosomes in the identification of the biomarkers using the super resolution microscopy. Next slide, please. So the student would grow different types of head and neck cancer cells and other primary oral epithelial cells in culture, and then isolate exosomes from these cell cultures and characterize them using nanoparticle tracking analysis, Western blotting, and different microscopy techniques. And he or she will also learn several associate techniques and other general lab practices. And yeah, with this, I would like to reiterate that our lab has experience and expertise in the field of head and neck cancers and exosomes, and can help and can support the student with all the guidance, training, and provide facilities required to successfully complete the project. Yeah, I would be available at 5 o'clock, from 5:30 today for the WebEx meeting, and I also you can email me anytime. Thank you. Thanks for being there.
Hello everyone, myself Tushar Ganjawala. I am a postdoc research fellow in Dr. Amanda Zacharias' lab in the division of developmental biology, and we study the wind signaling pathway in a simple animal model called C. elegans, which is a miniature worm. Next slide, please. So basically, we are interested in understanding how you go from a single fertilized egg develop into an adult worm. And a part of the process, as you can understand, that is about generating a lot of different types of cells, some of which will make intestine, the other will contribute to make neurons, for example, right? So how it goes from a single cell to the multiple cells and how the cells acquires their unique identity is based upon the external signals they receive. And wind signal is one of that. Okay, so in a picture you can see, without going into the much of the details, what the point I want to show is the genes that are dependent on the wind signals stays off when the wind signal is off, as you can see it on the left. That simply means that cell will not make those proteins that unless they get the green signal from the wind. Okay, next slide, please. And we studied this pathway in a limited worm, as you can see in a movie clip, which is about 1 millimeter long. And under the microscope, you can see this nicely developed adult worm, which have only 959 cells compared to the trillions of cells we possess. So it's relatively very easy to manage. And in spite of having such a low number of cells, they also have neurons, muscles, skin, intestine, just like we have it. And the same genes are involved in generating those cells. So it makes sense to study that relatively involved genes in a simple model. Next slide, please. And another very important characteristics about the C. elegans, Kyle, can you please click on the video, please? Is about they all have invariant cell lineages, meaning each C. elegans embryo develops in exact same way to generate from one cell to 959 cells. Means you can ideally compare one embryo movie with the other embryo. Is it playing?
No, I apologize. It's saying QuickTime is not available, so I don't think it will actually play.
That's fine. You can move it to the next slide, please. Let's see if that plays. Okay, so here you see the same, the movie where it's in color. I want to draw your attention. Here, each cell is labeled with a green fluorescent protein, and the gene of interest we want to study is labeled with a red fluorescence protein. So we can track the development of C. elegans embryo from one cell to an adult worm development. How we can take the embryo pictures every minute for about 5 to 6 hours under the confocal microscopy, and by labeling these genes, we can easily track and map the gene expressing in particular group of cells. In the video you just saw, you can one can definitely see that you can see more red cells on your left, meaning the gene of interest, which is labeled fluorescent protein, is only expressing on the A side, that is anterior side of the embryo. And those cells are believed to be developing, let's say, intestine. So we know that gene of interest is involved in the development of intestine. Next slide, please. So one of the wind-dependent gene we have identified, and we have also identified the group of cells it expresses, is NHR67, which is critical for the neuronal development. However, as you can see on the left schematic, that the red line, it presents where the NHR67 is expressed. The black represents the cells which it doesn't express. So how the cell decide in which cell is on and other cells is off, that is what we are interested in studying. So on your right schematic, you can see the where the right turn arrow indicates the start site for the gene reading, for the reading start site for the reading frame in a gene to make a protein. And left to that, there are sequences which are known to be involved in turning gene on and off. So we are interested in identifying those regions which are important in turning on and off the NHR67. One can systematically delete or put the extra piece of it and see what is the effect and expression and identify those regulatory sequence, which is called. Next slide, please. Most importantly, how and what can you contribute to these projects? Okay, so we use computational methods to predict those regulatory sequence, and then we use a molecular biology technique, including PCR subcloning, to test them in vivo. We also use a confocal microscopy. So here we present opportunity for you all to learn these molecular biology techniques or computational methods and execute this technique confidently after the project ends, and to apply them to answer a research question. What we expect from you is roughly about 12 hours a week over a 3 to 4 days. Okay, next slide, please. And I acknowledge all your attention, my PI, lab members, and currently we are 3 people working in a rotation to maintain the safety, social distancing. I also thank the UC Honor Program for providing this opportunity. Thank you all. You can contact me via email since I won't be available in a breakout session. Thank you.
Hi, everybody. My name is Hannah. I am a clinical research coordinator in the CLIP lab here at Children's. I'm presenting on behalf of Dr. Hopley. She's on a much-deserved vacation right now. But today I'll be sharing some information with you about exciting innovation opportunities for our study, or for her study on CRPS. Next slide, please. So first, let me start by describing a little bit about what we study. CRPS is a rare chronic pain disorder that severely impacts function in children. Our study aims to identify factors that can predict responsiveness to treatment in CRPS. Next slide, please. We use, we use 3 types of measurements to define predictive factors in CRPS. We use stimuli tools, like the one pictured on the left there, to measure changes in perception to sensations such as heat and pressure. And then we use surveys to see how changes in psychological and social factors. And then thirdly, we use MRI to define changes in brain activity and structures. Next slide, please. So due to COVID, we've made some changes to our testing protocols so that we can maximize everybody's safety. We've been using a 3D printer to create devices to help prevent cross-contamination between subjects and to aid in maintaining social distancing. And that's where we need some help. So that's where you guys would come in. Next slide, please. We are looking for a student with an interest in biomedical engineering to design and create 3D prints that aid in these COVID-related protocol changes. The goal is to deliver the stimuli to the patient without the study staff having to be close to the participant. Also, you will have the opportunity to think outside of the box, bring your own ideas, and to troubleshoot as you go. And depending on your interests, you will get to work with Dr. Hopley to explore and be trained on other aspects of research, including recruitment, testing, data analysis. And in best of all, you get to join our really fun lab team. It's full of other RaMP students, SURF students, postdocs, all from different backgrounds. And really, as a final note, I just like to throw this in. The great thing about clinical research is that you get to test really fun, big ideas while also making a direct impact of understanding treatments and the diagnosis of actual patients. So yeah, next slide, please. So if you guys are interested, you can join me at 5 o'clock in the breakout room, or this, and that's the passcode there. Thank you.
Hi, everybody. I'm a third-year graduate student in Dr. Rolf Stotman's lab. So we research the development of the brain and the face. And I'm working for dissertation research on this gene that's called SMPD4. So we're interested in this because we have 23 patients from different places all over the world who have mutations in this gene, and they all have microcephaly, which is small brain. They all have some other brain phenotypes, including cerebellar hypoplasia, which is an underdeveloped cerebellum, and simplified cortical gyration, which is improper brain folding. We primarily use mice to model these genes, and what we found already is that SMPD4 is expressed in the developing embryonic mouse brain. So this is the mouse model that I've been working with so far. It's basically a total deletion of SMPD4, the gene, and you can see in A and B that the mice that do survive that have this gene deleted are much smaller than their litter mates. The other thing is that most of the time they don't survive birth, so I've only been able to recover 2 adult mice from this line. And you can see that most of their brain, at least the frontal cortex, looks normal, but their cerebellums are underdeveloped and smaller in size. So that models the patient type pretty well. But what we're interested in now is understanding why the cortex of these mice look normal since the patients have microcephaly. And this is probably due to differences in human and mouse neurogenesis. So there are a lot of developmental processes that are the same between mouse and humans and how their brains develop. But humans have a greatly expanded cortex, and this is different from mice. So that's probably what's causing this difference in phenotype. Next slide, please. So what you would be doing in this lab is that I have been working on developing a line of mice that has SMPD4 knocked out, but only in the forebrain using a conditional allele. So what we're basically going to be doing is you're going to be looking to see if you delete this gene only in the forebrain. First of all, do the mice then survive because they're not dying from something else happening to them? And secondly, when or if they do survive, do they then have microcephaly unlike the other knockout line? So what you'll be doing specifically is learning skills like PCR and gel electrophoresis to identify mice that have the right genotype. You'll be dissecting the brains out from these mice and then sectioning those brains and doing some histology, which is the bottom pictures on the right. And then you'll be measuring these brains to figure out what exactly is going on with these mice. This is an in-person project, and it'll be 8 to 12 hours a week, and it can be whatever days you prefer. You'll just work it out with me. Anytime 9 to 5 on a weekday is fine. And I also have a Zoom session set up for after this if you have any additional questions. Thank you.
Hi, I am Bala Koritala. Our lab is mainly working on the circadian clock and obstructive sleep apnea. Actually, next slide. In brief, if we think about what is circadian clock, you see here we are living in the 24-hour rotation planet, experiencing light and dark cycles. And if we look at our activities, we are more active during the light phase and we sleep during the night. If we want to measure our activity and sleep patterns, you see that our activities are more during the light phase. That's why here you can see that the peak of, if you plot a graph, you can see that the peak of expression mainly happens during the light phase and then goes down during the dark. So this is the basic example of the behavioral rhythm you are seeing here. But these rhythms exist with heart rate, blood pressure, body temperature, hormones, and gene expression. Next slide. And obstructive sleep apnea, it's a sleeping disorder and tightly connected with the circadian, because the sleep is, if you see activity sleep cycles, then those are like circadian rhythms. Those are circadian rhythms. Here, the obstructive sleep apnea happens because when you have the throatal muscles are relaxed involuntarily, it blocks the air passage. That results in, you can see the hypoxic condition, less oxygen supply to the other tissues to function. The symptoms of this obstructive sleep apnea is poor sleep quality, snoring, increased heart rate, blood pressure, increased blood pressure, and cognitive impairment. These phenotypes are tightly linked with the circadian rhythms because we see that heart rate, blood pressure, and next. So here we are trying to measure that using the mice models, how these hypoxia impacts the tissue-specific circadian gene expression. And we keep the mice under hypoxia exposure, and we collect the different organs from the mice and check the gene expression profiles to see how does this hypoxia impact the circadian gene expression at the genome level. And next slide. So what you are going to learn from us, the importance of circadian medicine, and also you are going to analyze the big data sets, mainly the gene expression analysis against how these hypoxia impacts these gene expression analysis. And also you learn designing experiments, and you will acquire some presentation skills. Mostly now we are doing virtual because we are strictly not allowing the students inside the lab, but after if you feel interested, later we will figure out something to work in the lab. Thank you.
Shreyasi, are you here? Oh, you're muted.
Oh, I'm sorry. I'll start again. Hi, I'm Shreyasi. I'm a graduate student in Dr. Aaron Zorn's lab over at Cincinnati Children's at the Division of Developmental Biology, and I use genomics to study how stem cells choose their fate. Next slide, please. So I work with pluripotent stem cells, which are cells that can differentiate to any of the cell types in the human body. And the way that we get them to do that in the lab is by treating them with various combinations of signaling factors. Next, please. So when we think of a signal, we think of that if something turns on, then you have a specific set of genes that turn on. And if you don't have a signal, then genes turn off. But actually, it turns out that human development is a lot more complicated because cells receive a whole bunch of signals at the same time. So they must have some sort of mechanism to correctly interpret these signals and become the correct cell type at the right time in development. So this prevents, for example, lung cells from forming in the place of the liver. And this is, of course, very important in diseases like cancer, where if you have too much or too little of a signal, then cells behave abnormally. So what our research has found that these signals affect DNA in very specific ways. So next, please. So we think that by understanding how DNA, the structure, as well as the function of DNA is changing in response to the signals, we can understand how these cells are responding to various signals. Next slide, please. So our overall aim really is to use a wide range of genomic and next generation sequencing technologies to understand how different signals are changing DNA. And to do this, we ask where do these proteins bind on DNA, and how do they bind, and how do they alter DNA? And this allows us to then identify different genes that are being changed. So for a typical workflow of our experiments, we start with tissue culture, where we culture stem cells and we treat them with our favorite signaling factors. We then perform molecular biology techniques in order to characterize these different cell types. And we then perform a lot of high throughput DNA sequencing. And we perform a wide variety of these technologies to study exactly how DNA is changing in response to these signals. And we then have a bunch of computational pipelines to analyze these sequencing data. And this helps us to create networks to understand how these cells communicate with these signals. And of course, predict what would happen if a cell receives a particular signal. So because our workflow is intensive both computationally as well as experimentally, as a student in the lab, you will have the absolute flexibility to design the kind of project that you are interested in. So for example, you can choose to do, in the next slide, please. You can choose to do a very experimental or a wet lab project where you learn molecular biology techniques to understand how we characterize these stem cells. Or in the next slide, please, you can choose a computational project where you learn how to interpret these sequencing data and see how that relates back to basic biology. Or you can choose to do a hybrid project, which will have a little bit of both. But in all of these cases, what's important is that we will work together and we'll develop a project that you're interested in. And I think that this is a great example of the kind of, in the way in which biology is evolving further, where you have a lot of disciplines working together to understand these big questions about how stem cells respond in different conditions. So if you're interested, I have a Zoom room set up at 5, and I'm happy to answer your questions. And feel free to also contact me by email if you have any questions later on. Thanks.
I'm not able to hear you yet.
Oh,
there, that's working.
is it working?
Yes, it is.
Oh, I guess now it's fine. I'm using the airport. Hi, my name is Yaping Liu. I'm the assistant professor in Division of Human Genetics and Biomedical Informatics. So this project is actually impute cell-free DNA fragmentation pattern by deep learning approaches. My lab is a computational biology lab. So you will have the opportunity to work with me directly on this project. Next, please. So I'm maybe give you a little bit background about what is cell-free DNA and what is non-invasive prenatal test. So when cell is dying, the DNA will be fragmented into small pieces and released to the blood. And this DNA is outside of cell, so it's called cell-free DNA. So next, early in 1997, people already find out that in the fetus DNA is released to the mother's blood. And later, this technology was widely used in industry that called a non-invasive prenatal test. So people will use this test to know what is the genetic variation or genetic aberration in the fetus. So next. So in like 2012, people find out just by sequence this fetus, this circulating cell-free DNA in mother's blood, you are able to reconstruct the whole genome of fetus. Next, please. So this year, the ACOG, the American College of some Association, released the guideline that you recommend all of these cell-free DNA-based NIPT tests for all of pregnancy. That means like millions of these cell-free DNA whole genome sequencing will be generated for pregnant women. So next, please. So in my lab, one of the research directions we're focused on was using cell-free DNA to study the gene regulations, such as this like cell-free DNA from fetus. So you are able to know what is the gene regulation dynamics in early embryonic development. So there's a lot of study to study this like cell-free DNA epigenomics. They study the genomics called copy number variation, or study DNA methylation. What we are focused on was the fragmentation patterns. Next, please. So here's some project-related informations. Most of these NIPT test assay is performed at a very low coverage whole genome sequencing to save the cost. Or like this DNA is sequenced with the early pregnancy, so they have very little DNA from fetus. That means even you sequence them, you will still have a lot of genomic region that doesn't represent it in your sequencing results. That means a lot of missing data. Next. Next, please. So our goal is, if this data is missing, can we use the computational way to impute the complete pattern from this low coverage whole genome sequencing? So we are able to study gene regulation in millions of people, including pregnant women and fetus during this early development. Next. So what I want you to have is you need to be very familiar with Python and have some machine learning experience. And it would be great if you know some deep learning related technology. Next. So since all this work is computational work, there's no fixed time. As long as you can log into our server, you'll be able to work on it, and you will have opportunity to meet with me weekly, and also you can Slack with me like anytime you need help. Next is any next. Okay, yeah. So expected is since you are the only students working on this project and started this project, so you'll be co-author or even better authorship in the final manuscript. So it depends on your effort and contributions. So feel free to contact me after this presentation. I have a Zoom room in 5 o'clock, or you can search my name and find my email address, send me the email. Thank you.
Hi, everyone. My name is Shiba Brata Mukherjee, but you can call me Shib. I'm from Claire Chonier's lab in the Immunobiology Division in Cincinnati Children's Hospital, and we work on chorioamnionitis or in utero inflammation. Next slide, please. So what is in utero inflammation or chorioamnionitis? So it's an inflammation of the uterus, and it affects 10% of the term births and 50% of the very preterm births. So why do we care about them? We care about them because once affected, the fetus can have multiple morbidities like bronchopulmonary dysplasia, asthma, or cerebral palsy. Next. So as you can understand that this is a disease that affects the fetus, so we cannot study them directly in the human. So we have the opportunity to study them in a monkey model where we infect a pregnant monkey at almost 80% gestational age. And after the baby is delivered, then we study the disease. Next. So the reason of choosing monkeys is because they're very close to the human, as you can see in the pathology of the uterus, that they're very similar in how they look like. Next. So the background about this is in chorioamnionitis, we know that there is a plus of T cell differentiation and cytokine changes that happens, and you have multiple T cell changes. We are focused on studying the change in Tregs. Next. So why Tregs? Because these are important for maintaining the immune homeostasis, because if there is inflammation, the whole immune system is trying to protect your body. But what it's doing in the process is also harming some of the standby tissues or cells. So the regulatory T cells or Tregs suppress this activation by 4 different known pathways and try to bring in homeostasis to the process. So how do we study them? Next slide. Next. Next. So we learn about the basic characteristic of these cells by flow cytometry. Next. Next. And then we also have the capacity to manipulate these cells in vitro and study more about how their functionality takes place. Next slide, please. And then we can also understand how these cells interact with each other. Next. Next. So we do confocal microscopy where we study in real time how these cells are interacting with each other. Next. So far we have found some functionality changes and some surface marker changes in these cells. What that has done has opened options of studying these cells in greater details. So while in the lab, we have the opportunity to work on non-human primates and in a BSL2+ facility, and then learn high-end techniques of confocal microscopy, multi-parameter flow cytometry. And since it is at the initial stage or in the discovery component of the is very high, so we can work together to understand and get greater knowledge about these cells and how they function in the fetus, we would require an in-person presence in the lab. Twice a week for 12 hours would be fine. I don't have currently a Zoom meeting set up, but if you reach out to me via email, we can definitely have a Zoom call set up if you want to know something further about the project. Next. And I would really like to appreciate and thank you for the opportunity to listen to me, and thank you.
Hello, I'm Dr. Sumit Murad from Division of Orthopedic Surgery, Cincinnati Children's Hospital and UC College of Medicine. We work on 3D bioprinting of bone. The disease that we are focusing on is avascular necrosis, where the femoral head, the hip joint of the patient degrades because of the loss of blood supply to the femoral head. If this happens in the elderly patients, you can easily go for a hip replacement surgery and the quality of life is restored. But in pediatric patients, it is really tough because these kids are growing and you have to go for multiple hip replacement surgeries throughout their growing years. Their quality of life is totally deteriorated while there are multiple chances of infections all throughout. So next, to solve this, my slides are not basically correctly showing up, but I'll continue.
It may be a formatting issue. Yeah, I had to clear out the formatting so everyone's would be the same. But yeah, please continue.
Yeah, sure. To have a treatment for this disease, we are looking for an injectable treatment system that can be delivered with a minimal invasive surgery to the pediatric patient. And how we are developing this system is based on the tissue itself. Take bone and vascular tissue, we decellularize this. That means we take out the cells out of it, and now the remaining matrix, which is called decellularized matrix, acts as a reservoir of growth factors for differentiation of cells. Now we can use this extracellular matrix into different formats. We can make microspheres, embed it into microspheres made up of polylactic acid, or either we can re-mineralize the samples to develop a bioink. These two strategies are the main focus of our lab right now to treat this disease. When we develop the bioink next, we use different strategies. It's an interdisciplinary work that includes rheology, mechanical characterization, chemical characterization of the bioink, which later goes on to the biological characterization of the ink, where we test next. We put human mesenchymal stem cells into our bioink after we optimize our bioink. We have optimized our bioink, we put cells, and then with the live cells, we 3D print it into different structures. And then look after 28 days if our bioink is able to induce differentiation towards the osteogenic lineage or not. After this, we characterize the whole process of differentiation through RT-PCR, immunostaining, and other biological and molecular biology characterization techniques. So the whole work is quite interdisciplinary, which includes engineering and biology both. Next. So we have a lot of RaMP alumni from earlier years who are still continuing with us. So we are an interdisciplinary group including medical students, PhD students, postdocs, and surgeons. If you join for this project, you have to be open for working in different disciplines. You need to be open, and we need the student to be in person in our lab because this is a laboratory work. And you can connect with me after at 5 on my Zoom meeting room, as well as connect with me on my email. Thank you.
Hello, everyone. My name is Jiahong. I'm a research associate at the clinical mass spectrometry lab over here at Cincinnati Children's. Next, please. So we're using this technology called metabolomics approach for biomarker discovery. And we are interested in the metabolite biomarkers is because it's dynamic end products from the upstream genetic regulation and the protein bioreactions. So they are indicate what the current status of the biological system we are interested in, comparing to more upstream the genetic and the protein informations. So the early predictors by metabolites in various diseases attracted a lot of attention recently. So the technology we used in the mass spec lab is mass spectrometry. Next slide, please. So the samples can be biofluids, either the blood or urine, or can be tissue samples. After the processing or the extractions, we could be using two different kinds of techniques to measure the metabolites. One is the so-called untargeted, so we can looking at many metabolites at the same time. Those metabolites, including the small molecules, compared to the large molecules such as proteins, like amino acids, organic acids, and vitamins. So we're looking at many metabolites at the same time, and then we can discover the patterns and do some qualitative analysis for the biomarker discovery. And another approach is called targeted approach. So we focus a group of metabolites, and then we can accurately measure the concentration of that specific metabolites, and then use it as a biomarker for over-interested phenotypes. Next slide, please. So the two projects currently in our lab right now is one is very interested in to looking at how the dysbiosis of the gut microbiome and will affect the nutrition, especially the undernutrition in the children in the developing world. Is this so-called environmental enteric dysfunction, which is underlining due to the imbalance of the gut microbiota in the small intestine. It will affect the nutrient absorption and delay the children's growth. And another research project is currently going on is we are interested in looking at the genetic mutations and how that affects the cascade of the signaling pathways and affect the phenotypes. The two diseases we're looking at, one is neurofibromatosis type 1 F1 gene mutation. And so it's caused a neurological disorder. And another is a Fanconi anemia gene pathway deficiency, which also cause the pediatric cancer and other diseases for the children. Next slide, please. So the student working in this project is expected to be a combination of in-person and some virtual work, but it will depending on the children's pandemic policies. We expected at least some time in person, maybe 1 day per week, and in combination with some computational work. So you can have the whole picture of the project. And you will mainly working with me on the project, but there is a possibilities to working with other graduate students and undergraduate students in the lab. We are a clinical lab as well, so there is a very diverse background for the people working here. So you will definitely learn the biomarker discovery techniques and the related skills on that part. I wouldn't be able to stay late for today, but if you have any questions, please email me, and I will be happy to talk with you further. Thank you.
Hi, my name is Jay Pandavella. I'm a research fellow and also neural investigator by DOD at the Experimental Hematology and Cancer Biology at Cincinnati Children's. Next slide, please. So our lab works on a neurological disorder called neurofibromatosis type 1. So what it is it causes a benign tumor affecting the nervous system. It's been manifested by this a bunch of benign tumors called cutaneous neurofibromas and also plexiform neurofibromas. Most of the plexiform neurofibromas manifest at the younger age, and cutaneous neurofibromas as in adolescent and adults. So neurofibromatosis involves this signaling cascade called the RAS-MAPK kinase pathway that involved in the regulation of cells, cell growth, and also cell survival. So in neurofibromatosis type 1, there's a mutation of the NF1 gene that codes for the protein neurofibromin, which acts as a brake for the activation of RAS. So if you have this mutation, you have a loss of function of the neurofibromin, and this leads to the hyperproliferation and the uncontrolled survival of the cells. Next, please. Next, go back. And where this mutation occurs is in the Schwann cell lineage. And what happens is when you have the neurofibromin or the NF1 gene mutation, you have the hyperproliferation of the Schwann cells, and this would undergo subsequent events involving inflammation, and you have a maintained inflammatory response that would contribute to inflammation development of the tumor. Next, please. So we have a mouse model in the lab where we've conditionally knocked out the NF1 gene, or we specifically target the NF1 knockout in the Schwann cell lineage. And on this model has been a great tool in understanding the disease. So it has similar features of what we see in humans. And this has led to the success of knowing a target using a MEK inhibitor that has been recently been approved by the FDA. Next, please. Okay. And in addition, this model has also go back, please. So this model has also shown that it mimics a precutaneous features. So this mouse has shown that the skin sort of have a dense nerve innervation of nerve fibers. This is similarly found in humans, and these nerves are also expressing or produces receptors that are responsible for nerve growth and also nerve survival or neuronal survival. What we call is the glial-derived neurotrophic factors, and these are a family of growth factors. Next slide, please. So the project that the honors student will be involved in together with me will have to define the role of this growth factor, the GDNF growth factor, and how we mediate this neuronal and Schwann cell crosstalk. In vitro, they will be involving primary cells or some culture immortalized Schwann cells. And also we would have to test, look into what would the effect of MEK inhibition in this interaction affecting the growth factor signaling in vivo. So what would the RaMP honors student will be expecting from us is learning a bunch of lab techniques and of course, understanding the Schwann cell biology. So you're going to be learning a lot of the mouse husbandry associated skills, gel electrophoresis, growing up primary cell cultures, imaging techniques, confocal microscopy, and electron microscopy. What we want from the honors student is that consistent enthusiasm. So, you have something to look forward to in a day-to-day basis, so that, like the project will get, especially if you get good data, and you will have something to look forward to. And also, you'll have to be a team player. So we are a multidisciplinary team. So we've got clinicians, bioinformaticians. We have PhD students and rotational students as well, where you could seek help. So you're expected to be in the lab, in particular in the first couple of weeks, where you're going to be familiarizing the day-to-day workout in the lab, and that could be changed and be flexible as we go along in this project. Thank you. By the way, I'll also have a Zoom link that I've sent to Kyle, so I'm available after 5 if you want to talk about or have any questions. Thanks.
Oh, hello, everyone. So I'm Luis Keme from the Pain Management Lab at Cincinnati Children's Hospital. And recently, our lab started this brand new research avenue looking at the link between stress and the development of chronic pain. And this is important because pain patients with chronic pain conditions, things like fibromyalgia, frequently present with increased stress and anxiety. And in general, it's such an stress is such an important modifier of the pain perception experience that it can be used as a predictor of post-surgical pain outcomes. So kids that are in more stress, they usually experience more pain after their surgeries. Also, kids that have a lot of stress in early life, like kids in the neonatal intensive care unit, they tend to experience pain with more intensity across lifespan. So we know that stress can extremely affect how we feel pain, but we don't really know how. And we're trying to look at the role in stress of stress in activating the immune system and how this will influence the perception of pain at the level of the peripheral nervous system. Next, please. So this is where you guys would come into the project at the lab. Basically, we have developed this model of stress by using loss of enrichment. So what we do is we put mice in an extremely large cage with a lot of toys, give them an enriched environment that they can enjoy. And after 3 weeks of living in this environment, we remove them and we house them alone. So imagine if we put a mouse in a lockdown environment like we all experienced back in March when the pandemic started. You basically lose all of your motivations and your life feels a lot less fulfilling as it used to. But we cannot ask a mouse how much pain it's experiencing or how much stress they are having. So we have developed this AI-assisted motion tracking tools that allow us to detect very small changes in the mouse behavior that can point us towards if a mouse is experiencing pain or if the mouse is experiencing increased stress. We combine this with multiple techniques that involve also electrophysiology of the sensory neurons in the peripheral nervous systems and immunofluorescence techniques that allow us to track this little red dots that you're going to see in your lower right screen, which is basically immune cells migrating into the muscle that we believe might be affecting how the peripheral nerves are feeling and perceiving pain. Next, please. So what I would be expecting of you if you join our lab, it's basically work that will be mostly in person, since we're also working with a computer model that part of the coding, if you're familiar with that, would be done remotely, but it also involves a little bit of in-house work. The time of the work, it's something between 2 to 3 days per week, about 8 to 12 hours, depending on how involved you become in the project. And I'm definitely interested in working with someone that would like to stay for the summer on top of doing the spring semester. If you want to talk more details about the project or how you could be involved with it, I have a breakout room at 5 p.m. Please join it. Thank you.
Hello, I'm Mark. I'm a researcher at Children's, where I'm interested in studying how time of day impacts biology and health. I'm a postdoc fellow in the Hoganish lab, so my own mentors are John Hoganish and others in that lab. Next slide, please. So I know I've only got a couple minutes here, but I couldn't resist showing you what I think is this pretty cool reminder of how our bodies organize themselves around time. So this plot is actually called an actogram. It's from a newborn. So to orient you, each revolution, each circle is 1 day. It starts in the inner circle at birth and spins outward as time goes on, starting at birth and kind of spinning through to, I think, about 5 or 6 months here. And so times when this baby was sleeping are in blue, when the baby's awake are in yellow. So at the outset, in the very inner circle, you can see newborn sleep is pretty oblivious to time of day. Then this baby actually goes nocturnal for a few weeks. And then finally, there's this kind of amazing transition where sleep becomes consolidated to the overnight time. And as a new parent, it's pretty heavenly. So this is a classic example of a circadian or 24-hour rhythm. Next slide, please. But it isn't, sleep isn't the only time of day regulated process. In fact, we now know that expression of about half of our genes and much of our physiologic functions fluctuate over the course of a day. And so our lab and my particular interests are centered around characterizing these rhythms and understanding the ways in which they may matter for health and for medicine. Next slide. And so my approach is computational and takes advantage or aims to take advantage of the huge amounts of data that are generated via just everyday healthcare. So every time you or I go to the doctor, we contribute data, and most of that is never used by anybody. And so this RaMP project, if you are interested, involves searching for time of day signal in these massive human health data sets. Next slide. And specifically, you'll help to explore how clinical lab tests, so the things measured every time we go to the doctors and have blood work, blood is drawn or urine tests, all those things that are measured, exploring how they vary by time of day. And the project does not require that you have a programming background at all. If you've got one, that's great. Otherwise, you'll learn. So my goal, I think, is to help teach the tools for data analysis and data visualization. Hopefully, that will be useful to you, well beyond this project. So thanks for listening. I hope to hear from you. I'm unfortunately not available after this, but please feel free to email me anytime if you'd like if you're interested and would like to learn a bit more. Thank you.
Hi, everyone. My name is Sandra, and I'm a postdoc in Elisa Boscolo's lab at Cincinnati Children's Hospital Medical Center. And our research focuses on venous malformation, and venous malformation is a developmental defect of the vasculature that is mainly characterized by massively enlarged veins, and you can see some examples of this disease in these images. And these vascular lesions are extremely painful and can be quite disfiguring for the child, and they grow with the patient and they never spontaneously regress. We do know the genetic mutations that are causing this disease, but
Relatively little is known about the molecular and cellular mechanisms that underlie the disease, and my project focuses on how the cells that carry the mutation, so the mutant cells, are communicating with the healthy wild-type endothelial cells. Next slide, please. And I have discovered that these mutant cells are secreting high levels of repulsive proteins that are called semaphorin 3A and semaphorin 3F. And I'm currently investigating the effects that these repulsive signals may have on the healthy endothelial cells. So in the bottom figure, you can see one of our model systems that we use in our lab. It is a three-dimensional model of vascular lumen formation. And so therefore, we coat microcarrier beads with healthy or mutant endothelial cells and then embed them into a fibrin gel. And after a few days, these cells start to sprout and form lumens. And we can see that the mutant cells really form very large vascular structures compared to the normal endothelial cells, and therefore they really mimic the disease in the dish. So if you decide to join our lab, you will be learning different basic lab techniques. For example, you will help with cell cultures, you will learn how to measure RNA and proteins. We also do a lot of immunofluorescence staining and histology, and you will be also helping to genotype our mouse models and, of course, learn how to analyze and present data. So I'm very flexible with what days you decide to come in. It is just important that we communicate in advance, and I will be available for further questions between 5 and 6 in the Zoom meeting, and I believe you have all received the link, and I hope to see you there. Thank you.
And yeah, just a quick update for any students who may have joined us late. If you have not gone onto the RaMP website and looked at the RaMP mentor project descriptions, that is the document that several of the mentors have referenced when they've talked about joining their Zoom links or their WebEx links if they're available to, this evening at 5 p.m. So make sure you check that out. And with that, we will continue on with our presentations.
Hello, I am Brittany Smith. I am a postdoc with Dr. Teresa Reyes in the UC Department of Pharmacology and Systems Physiology. We are located at the UC Reading campus, or Metabolic Disease Institute. So that's right off of Ronald Reagan Highway and 75. You get free parking there. It's a great place to be, but it is a different location from the med campus and children's. So keep that in mind. And in our lab, and the project that I'm working on and looking for a student to work on is looking at the maternal opioid epidemic, which is paralleling the opioid epidemic in the general population, where we see this dramatic increase in opioid use by pregnant women and a subsequent increase in the hospital admissions for infants that are undergoing this neonatal abstinence syndrome, where they're born suffering withdrawal from the opioid drug. And so we have this dramatic increase where this is an older estimate. Estimates are even higher now. Every 25 minutes, a baby is born suffering from opioid withdrawal. So super relevant public health crisis we're tapping into in our lab. And in the tri-state area, you can see we're in a hot spot for these neonatal abstinence syndrome cases. And so in our lab, we are a molecular and behavioral neuroscience lab. So we're interested in understanding the cognitive consequences for the children that were exposed prenatally to these opioid drugs, because some of the clinical literature has shown that, you can advance the slide. The clinical literature has shown that in humans, there are cognitive delays and impairments in these offspring. They're inattentive, hyperactive. They suffer from ADHD, conduct disorder. They have disruptive behavior, impaired inhibitory control. They struggle with poor planning, adaptability, and short-term memory. So a huge battery of behavioral deficits in these children are emerging in the clinical literature. And so to understand that more, our lab is using a mouse model of this. So that's what the project would entail, is using a mouse model of maternal opioid exposure
to understand the cognitive consequences for the offspring. Next slide. And what we have grown to appreciate and understand and want to incorporate in our model is the complexity of the opioid using population. So the opiates are not the only insult on board. There's also very high comorbidity between substance use disorder and mental health disorders, and comorbid mental health disorders and very stressful life environments. So we're trying to model that next in our model, and this is the experiment that we would be using a student to help with our project, and they could even develop their own sub-project within this larger study. Next slide. And to model this early life stressful environment, we are using a limited bedding model plus minus the opioids on board. So you can see in the control animals are given ample bedding, and if they're stressed, they're given this mesh grate and reduced number of bedding to induce a stress phenotype in the context of opioid drugs or not. So we think that like the double hit of stress and opioids may precipitate worse cognitive outcomes. And the student commitment on this would be to help breed and study this mouse cohort. This would be the first time doing this study, so we would have very detailed assessment of these mice and how they do throughout pregnancy, how the offspring do, track their body weights, track pregnancy outcomes. We would look at maternal behavior with the pups, and then also track the offspring as they age. Depending on the commitment in the lab for how long you could stay, you may or may not see some of the behavioral testing in the offspring, but we would definitely be doing some molecular assessments of the offspring in this model. Larger blocks of time are preferred but not required. We're available during normal business hours from 9 a.m. to 5 p.m. This would be majority in person given the animal model. There will be bits of remote work that we can sprinkle in here and there, but mostly in person. And I am available afterwards from 5 to 5:30 for a breakout session that the link is posted.
All right, good afternoon, everyone. So my name is Elon. I'm a junior faculty at the radiology department in Cincinnati Children's. So I lead most of the deep learning-based research efforts within the department, and our core functions are, first of all, to anyone who doesn't know what deep learning is, it's a class of algorithms that comes under machine learning and a broader umbrella of artificial intelligence. To cite a general example, you would have all heard about Tesla's autopilot, which helps them, which helps their cars drive autonomously, and that's primarily driven by deep learning models. Similarly, if you use any imaging app, Instagram, Snapchat, all the filters are all based on deep learning. So deep learning has done wonders to the computer vision community. It has revolutionized the field in many ways, and we use that in radiology to help our radiologists make better decisions, to improve the workflow for patients and staff, and to enable research applications that wouldn't be possible without deep learning and the automation it brings to the pipeline. We also, once we develop the models, we also want to validate it so it's safe and effective for clinical use. And we have a GPU server, which we use to train our deep learning models, and there are other folks in the department who want to use it, so we manage the server for them. Next slide, please. So here I have 3 examples to just give you a taste of what we do. The first example is a deep learning model for 3D MRI liver segmentation for kids. So right now the process is a kid comes and gets an MRI scan to make a diagnosis of liver disease. So there is a person who sits and basically manually contours the liver before the radiologist looks at the image. So we developed this model to replace the person and make it more standardized, and the model works at a 94% accuracy with respect to human, and it takes only like
a couple of seconds to do the whole liver volume. Similarly, we have another project where we are trying to quantify the nodes for a disease called sarcopenia, which is associated with muscle loss. So here we are looking at a CT data set where a model goes and looks at the sagittal view of the CT scan, which is the first image here on the lower right. And it tries to find the L3 vertebral level automatically, as pointed by the arrow there. And then a second model goes in and segments out the skeletal muscle area. And the third figure here is a model which was used for workflow improvement in the emergency department. So if a kid comes with an airway infection, or if they have some foreign bodies in their airway, they might have swallowed, we usually take a radiograph of the kid. And oftentimes what happens is the kid has to wait within the radiology room until the radiologist looks at the image and says, hey, this image is good, and I can make the diagnosis of it. If not, they'll have to get a repeated scan. So this process usually causes a delay of 15 to 20 minutes. So we developed a model to see if we could introduce a deep learning based pipeline that can detect the scan as soon as it's done to tell the technologist if it's good enough or not for diagnosis. And we validated it against radiologist performance, and we found that it works as good as a radiologist, in some cases even better. And this is a post-validation image where something called a class activation map, where we evaluate where the model is actually looking at, because we just want to make sure the model is actually looking at the same anatomy as a radiologist would. So if you notice it here, in some cases where there are some artifacts, the model looks at some of the line region here instead of focusing on the airway itself. So we're currently working on methods to integrate it in the clinical workflow and see how well it does, and also to improve some of these artifacts. Next slide, please. All right, so if you join our lab, you can expect to learn the basic components of a deep learning pipeline for imaging. And you will also learn coding with Python and get to experience using some of the deep learning libraries like TensorFlow and Keras. Also, depending on the project, you might have to do some of the manual segmentation to compare your model's performance against a manual human observer. So you might learn some of the tools that we use to actually create the manual segmentations, and you might even work with a radiologist who teach you how to go about contouring the anatomy you're interested in. Also, you'd get to use some of the advanced NVIDIA GPUs we have to train deep learning models. Next slide, please. All right, so what we expect from you is ideally, if you have some kind of coding experience, that will be great. But even if you have some interest in it, I think we can work out and have you go through some tutorials and make you ready for it. And you should be interested in troubleshooting. Basically, software development involves a lot of bugs, and you should have the mentality to accept the challenge and go and look for the solutions. So you should know how to use Google search effectively, because most of the solutions are out there. And since this is a rapidly evolving field, there is a new solution for a problem every 6 months. The solution changes. There is a faster solution or a better solution. So you should be able to use Google search effectively to find the most optimal solution at any given point of time. And we expect you to have some proper communication in terms of emails and commitment to meet deadlines. Since this is all software-based, you can work in your convenient time from your home, most likely. We expect you to put in anywhere between 12 to 15 hours a week. And most of these projects are at different stages. So depending on the work, you'd likely be part of some journal articles that we produce from these works. So I'm not unfortunately available for the Zoom call after this presentation. So you can contact me by email, and I'm usually prompt at responding. So also we can take up to 2 students. We have multiple projects and we can easily have 2 students. Thanks.
All right. Hello, everyone. And I'm Durgesh. I'm a research instructor in neurology, and I work in Waltz Lab. So in our lab, we're interested in understanding the mechanism of neuronal hyperexcitability, or a network which is hyperexcitable. And we study epilepsy and autism, which are the disorders characterized by increased neuronal activity in the brain, which leads to excitability, and there is behavior and cognitive deficit. So as you can see here, in a normal human brain, you will have normal functioning neurons, or a happy neuron, you can say, and this is, I'm representing a normal person as well as a child. So because we study epilepsy and autism. So however, in case of, for example, epilepsy, there will be an increase, there is an abnormal behavior which will be associated with the increased abnormal mental activity or brain activity. And you can see here at the bottom, there are neurons which are firing more than usual, which is abnormal firing, or increased firing as in increased hyperexcitability. So the reason epilepsy matters because there's 65 people around the world which are affected by this disorder, and autism has even larger numbers. And then available treatment for both the disorder does not work in 1/3 of the patient. And there is no potential cure. There is no seizure man. There is no better therapeutic. There are therapeutics, but there are no cure for these disorders. But there are ways to study this disorder and then also to target and understand why this happens. So for that, you study ion channels or abnormal mutation which occurs in these disorders. And in our lab, we study one ion channel, which is Kv4.2, which is involved in mediating this hyperexcitability, and another protein, CNTNAP2, which is also associated with autism. And Kv4.2 has been associated with both autism and epilepsy. Next slide, please. So you will be studying, if you are in the lab, you will be studying these 2 proteins, Kv4.2 and CNTNAP2, using the mouse models. So we have mouse models which are the knockout as well as the heterozygous mice, which you will study. So there you will do the estimation. So we have different projects in the lab, and we have published some projects as well. But for the initial phase, you will be involved in training with the techniques. So for example, you will do immunohistochemistry, which is just a general antigen antibody reaction through which you can detect the presence of those presence or absence or an effect of treatment on those protein levels within the mouse brain using this confocal imaging. And you would be part of these general experiments, which are general day-to-day lab experiments called genotyping, which is to find from a colony which mouse has a knockout, which is a knockout for Kv4.2. So you can study the absence of Kv4.2, and similarly, if there is a mouse which is knockout for CNTNAP2, so you can study the absence of that gene and its effect. For example, these knockouts usually display seizures. So here, there comes this part. So this is an EEG trace. And in this case, if there is an only online option, this is possible to do it online too. So we can talk as if you choose to be in the lab. So through this, you can actually look into this EEG analysis as in the brain signals which we get from this mice, and I do all those brain surgeries. And so you can look into the spikes, so you can count how this model is generating spikes. You can count how is it generating seizures, and then in EEG oscillations or the waveforms, which gives an idea of how the network is hyperexcitable, or what is happening to the network due to the lack of this 2 proteins, or if there is a heterozygous mice, which are kind of partially expresses these proteins as well. And then we can find therapies to guide. So here are some of the images from the lab. So I have had students before. So Alex was my student, and Andrew, who's doing a PhD at UC, and Andrew is now in med school at UC. Both of them were with me for like 3.5 to 4 years, and we have published 2 papers with them, and they have presented at all the opportunities provided. Currently, I have 2 students, Rishav and Meghna. And you can see in the pictures, these are our senior lab members, Dr. Gross, who I work with, Angela, who will talk to you in a moment. And this is just a general lab image from, I think, 2 weeks before. More importantly, I'm looking for students who are interested in basic research, and also long-term students are more preferred, so I can guide them and they can be part of papers, and they can be part of different projects too, because like what Alex and Andrew has done in the lab too. That's all. And contact me if you have any questions. I won't be available, but we can schedule one-on-one meetings, and
Yeah, let me know if you have any queries through email. But yeah, thanks for listening. Have a nice day.
Hi, my name is Marina. I work in the Division of Pulmonary Biology at Children's Hospital. Next slide, please. As you know, our division is focused on the role of lung development and disease. As you know, lung, which is a respiratory organ, which comprises of different compartments, starting from trachea, it compartmentalized to bronchi and bronchioles to the alveoli. Alveoli is the functional unit of the lung where the functional gas exchange occurs. So alveoli, which comprises of multiple cell types, both epithelial cells and mesenchymal cells and endothelial cells. So epithelial cells, that is a functional cell types, are responsible for the initial gas exchange, but the fibroblasts, which are the stromal cells, which support the epithelial cells for the proper gas exchange process. So in our lab, we are interested in studying the role of cell-cell interactions and see how these interactions help to maintain a proper equilibrium in the alveoli, which helps in the proper gas exchange process. So from the alveoli, we are interested in fibroblasts. They are the stromal cells, and there are different types of fibroblasts which are present in the alveoli, like myo, matrix, and lipofibroblasts. Each of them has different roles in different stages of lung development, and which is important for the proper development of the lung. But here we focus on the matrix fibroblasts, which are supposed to be the beneficial fibroblasts, and nothing is known much about matrix fibroblasts. So during the lung development, how these matrix fibroblasts crosstalk with the epithelial cells for a proper gas exchange process. And here we zoom into the gene, which is the GATA6, which is recent studies from the computational analysis showed that GATA6 can regulate this matrix fibroblast phenotypes. So we are studying how GATA6 regulates this matrix fibroblast for helping the epithelial cells to function properly for a proper gas exchange. So here we use both mouse models and cell culture systems to study this. And next slide, please. So mainly the role and responsibilities of the students who will be joining would be like they will definitely learn how we design the project and how we, from an organ to tissue to cell to the gene. That's what I just explained, is how these cellular interactions are really important to carry it forward for understanding the basic developmental mechanisms. And also they will definitely learn the basic laboratory practices. And experiment wise, I'm planning for both analyzing the gene and protein expressions by qPCR and immunofluorescence respectively. So they will definitely learn to quantify or analyze the data from qPCR, and also they will learn how to process the tissues for immunofluorescence. So all these are wet lab bench work. So I would expect we in person only, mostly 15 hours per week, but the timing, the student can decide the time and they can split the time accordingly. So I will be available after 5. So if you have any questions, I'm happy to answer. Thank you.
And I'm going to jump on very quickly here and just remind students that your mentors are not going to be going anywhere for those 5 p.m. meetings. We only have 2 presenters left, so please make sure you are watching their presentations, and we'll probably be a tiny bit after 5, but you'll still be able to connect with them.
All right, I guess I can take over now. Let's see, sorry. All right, so my name is Angela White. I am a postdoc in the Gross Lab, so that's the same lab that Durgesh is in. And so I study autism spectrum disorders, which we know affect 1 in 54 children. And what you're looking at to the left of your screen is a large-scale study that was done where scientists were interested in seeing what genes really drive autism. And so this is a diagram of the genes that they found to be commonly mutated. And importantly, our favorite gene is up there in the beautiful red star, and that's P10. Next, please. Thank you.
So P10, interestingly, is one of the most mutated genes in all of cancer. And so this was a really interesting finding because before this work, it really had not been connected to anything but cancer. So mutations in P10 alter the PI3 kinase pathway. So when you alter PI3 kinase, you get increases in cell size and changes in cell cycle, increases in cell cycle is probably the better way to put that. And so when you actually, we went back and started looking at what was known from cancer research about P10 deficient cancers. And that's where we got a lot of our ideas that we're trying to assess now to see if they are relevant in P10 deficient autism spectrum disorders. And importantly, one thing that has been found in P10 deficient cancer is that you can inhibit one of the catalytic subunits of PI3 kinase, there are 4 total, called P110 beta, and you can actually stop tumor growth. And so the big question that I have in my project is, can we also inhibit P110 beta in P10 deficient autism spectrum disorders and reverse some of the phenotypes? And so next, please. Thank you. And so essentially to answer these questions, we use 2 different types of models. We use tissue culture specifically, we use primary neurons, and then we use a mouse model as well. And this is a genetically engineered mouse model where we're able to selectively delete P10 in a subset of neurons. Next, please. Thank you. And so in the tissue culture, essentially, what we're assessing, we utilize a couple of different techniques where we're able to knock down P10 in these neurons. And so what we're assessing are changes in the PI3 kinase pathway. And then in our mouse model, what we're assessing, we also assess cell signaling there as well. But interestingly, we know that most of these mouse models have seizure phenotypes. And so we use P110 beta inhibitors to assess the seizure phenotype, and then we also can assess a behavior. And so in terms of what we are looking for a student to do, we're very flexible this time around because of the pandemic. Next, please. And in terms of hours, what I have on this slide is very minimal, but that is because of the pandemic. We would really like to have a student around more. 3 days a week would really be wonderful if possible. And 2 to 3 hours is minimal. If we could do more, we would love to. And we, several projects that I have would have students within the lab actually at the bench doing work. And some of the things that I'm interested in having a student doing is helping with protein assessment as well as doing some of the genotyping for the mouse models. However, because of the pandemic, I do have a couple of projects that could be done virtually. So that's all up to the individual student and what they're going to be comfortable with. And as Durgesh has already told you, we've had a lot of students come through who have had multiple publications, and we would love to keep that tradition going. And unfortunately, I will not be able to stick around after this meeting, but I have my email address at the bottom, and I would be more than happy to schedule a virtual meeting or talk via email with anyone that has questions. Thank you.
Hi everyone, this is Danny. Well, thanks Kyle for the invitation and set this up. So I'm assistant professor of College of Medicine, Biomedical Informatics, and my lab innovates clinical data capture and use. ICDCU for short is, I would say, a very different lab from most of the labs in the College of Medicine, because we're working on the combination of technology, information, and clinical problems. And we care more individuals, teams, and organizations than, you know, genes and then cells and all that stuff that I'm not really familiar with. So the idea here was to develop an information system and taking care of data from either structure on structure. Structural data here means, you know, the data can be organized and managed in relational databases. Unstructured data basically means notes or any narratives. And through data analytics, machine learning,
NLP, natural language processing, and we're able to develop a system to, present information in an interactive visualization or dashboard. And then that will often involve end users, primarily clinicians at UC Health or Cincinnati Children's. So my lab is pretty much problem focused, and very often we have clinician collaborators that help us refine the problems and then define the scope of the project, and then also evaluate whether the system is effective or not. And then in academic hospitals, there are three missions. They do care delivery, clinical research. Many of you have seen projects like that today, and then also medical education. 50% of my time probably, or projects probably devoted to care delivery, and the other half going to clinical research and then medical education. So just to clarify, for clinical education, I don't do much about the bioinformatics, if you will. It's more about infrastructure development. So these are the buzzwords. And then if you were to join the lab, I'm recruiting 4 students this year, 2 for the research team and 2 for the tech team. For the research team, you will be working on clinical workflow analyses, which employs mixed method research. The other side, if you're a tech student, you're working on more on the NLP and text mining stuff. Next slide, please. So basically, I think these are the sort of the formula in my lab. I think a commitment is really something I'm looking for, in addition to the great motivation that you really want to do research as part of your priority. I know for undergraduate students, based on my experience, there are a lot of competing events. So if you really can, I'm not going to compete with exams on the or requirements, but other than that, I will hope really be top of the agenda. And then I really focused on the communication as well, close communication, tracking all the stuff. I do that myself, but also put you with teams in other students. I have about 15 lab students, many of them undergrads. So you will work in smaller groups. Next slide, please. So for the 2 research interns I would like to recruit, I'm going to show you some of the achievements the students have been developing. I have 2 students per semester per year, and then most of them are medical sciences majors. I'm not going to go through details because I'm hosting the meet and greet after this. So really the requirement is, again, working on clinical workflow analyses, conducting structured interviews with clinicians, doing more qualitative aspect of things. If you're a new Python, that's great. If you don't, then we have the training material that will help you go through that. And after that, you can start to do some data manipulation analysis tasks. Next slide, please. So Scott Vandermeer, which is the most senior student in my lab, I did set up a fellowships in my lab so people can get paid through that fellowship position without clocking hours. We can talk about that in detail if you're interested in my meet and greet. And then Scott have over the year author abstract, and then also a second author journal paper at the end. He is focused on a resident dashboard in the medical education to create a visualization and refine a current dashboard with internal medicine. I'm gonna skip the next 4 slides because each of them describes the individual achievement of the lab members. And these are all medical students, science students. If you're interested, take a look at them. For the text interns, I'm also recruit 2 students as well this year. Next slide, please. This is the research pipeline is more developed. The tech pipeline is not. So I'm trying to develop that as well. And again, I'm looking for a longer term relationship. So after RaMP, I have a whole plan that can help you succeed through the 4 years of your undergraduate study. So if you're interested in that, definitely join the meet and greet. I can explain more. The tech intern, on the other hand, is not that developed, but I try to combine this RaMP with co-op requirements, especially there's a research EEP in co-op that people can combined. So that's my focus this year. So for the tech intern, really, either you are experienced in web development, especially Python, Flask, all the JavaScript, all the interactive stuff, or you are more backend development, still Python, but more scikit-learn and then machine learning data mining type of experience. If you have to know Python at least, otherwise it's hard to start to build upon things based on your skills. So I'm looking for more in this category, computer science or information technology students. So I have some examples here, but I want to highlight Piyush, who received a CES Tribunal scholarship based on the work with me using his research EEP experience in January. So I think we can tap into that and then continue that project if we're interested to join. Next slide. Okay, so this is an overview of the plan. As I mentioned, it's a whole training, and then we do have a interview process. So I'm gonna talk about that in detail in the next 5 minutes, after 5 minutes, and then join my room if you're interested in. But before you talk to me further, you will talk to Catherine, one of the senior juniors in the lab, who is the sort of the chair of the committee, if you will, for this recruitment effort this year. All right, I think that's all I want to share. Very pleased to meet you all here, and then all my lab work can be done remotely. If you're interested in this type of work, definitely, again, join the meet and greet. Thank you.
So I would like to thank once again all of our mentors who have joined us today, who are those of you who are still on the call. Students, thank you for your time and attention. I will have a video, because this is being recorded, I'll have a video of this presentation posted probably on Monday, because it takes a little bit of time to process and get up on our website. Cindy, any final words from you before these students? Mm-hmm.
Yeah, one thing, remember when you're interviewing mentors, there's a difference in requirement for working at Children's versus working at the College of Medicine. If you're going to be working at the College of Medicine, your current badge works just fine. If you're working at Children's, you need to get a Children's non-employee badge. This requires extra work on your part and on the mentor's part. So you need to have an updated two-part TB test, an updated flu shot, and you need to provide all your vaccination records. So if you're a pre-med, you probably got all that stuff under control already, but if you're not, you may need to start thinking about that. And that's why we want to make sure your match gets made before you go home for holidays. I know people are going home earlier now, so we want to make sure that we have good contact information for you so that you can get all that information to us and get your badge when you come back from Christmas break. That's all I have to say. Have a great evening, everybody. Good luck with your matching. Bye-bye.
Thank you, Cindy. And once again, thank you, students, for your attention. Email me if you do run into any difficulties throughout this process. But all the information you need should be up on the website. So go join these breakout sessions, talk to these mentors. Remember, you can visit multiple breakout sessions too. Most mentors are available until 5:30. And I think I heard a couple individuals state even 6 o'clock. So take care, everyone. Stay safe, stay healthy, and look forward to seeing how you match with your tours this year.